because I want to promote a positive environment. You're not recording all this, are you? Yeah, that's fine. I just have to be.
Share it
I
on the
have to
discussion.
behave now. Okay, so let's talk about this stuff, and I want to tell you what my expectations are. So, when we look at a slide, because there's questions on here, your exam will not have questions on them. I'm just going to pop up an image and I'm just going to ask you what comes to my mind. That way, no practical is the same. Okay? So the first thing I want you to consider is I want you to look at that, and if you see red and purple, what do you think?
Gram positive.
Well, no, not gram positive. That is gram positive, but you always think gram stain. So you think gram stain. If you see red or purple, you think gram stain immediately, unless I tell you differently. Okay? So now, if we know this is a gram stain, what is the gram reaction at the pointer? Gram positive. You can say gram positive, or you can put GP, but you cannot put gram with a plus sign. That's not acceptable. Okay? Good? All right. I know. I'm just. Yeah. It's not acceptable. It's not what is found in science. Okay. So what's the difference between a gram positive and a gram negative cell? There are many. I just need one. And you would say gram positive have a thick peptidoglycan. Like it. What else could you say? Gram neg have an LPS. I like it. Gram negative have a smaller peptidoglycan. Like it. Okay? There's a lot of them. I just need one. Okay. Identify the arrangement. Yeah, I would accept that too for gram neg. Yes? Those are strepto. And they are coxy. So if some people look at this, they might think it's bacilli. And if nobody asks for clarification, because you are allowed to raise your hand and say, Provi, can you tell me what I'm looking at? And I'll say they're looking at the little round things. I'm not going to say they're coxy, but I'm going to say the little round things. But if nobody asks for clarification and I don't mention it, I will accept both coxy and bacilli. I am very amenable to your evaluation because these things are distorted, and I know they are. Okay? But the answer to the question, what is the arrangement, is strepto or chains. Those are the only ones that are acceptable. Okay? Name the counter stain used in the gram stain. The counter stain is the second stain. Huh? Safranin. I don't count off for spelling, but if you mean safranin and you spell it saffron, that's a whole that's something completely different. Saffron is a spice. So words have meaning. Okay? Good? Anybody have questions? By the way, I'm really craving Indian food right now since I said safranin. Here we go. So, if you see purple and red, what? Gram stain. If you see green and red, endospore. And so that's the way I want you to think about it. I want your mind to focus on what you're looking at and come up with an idea of what you're looking at immediately. So you look at this, oh, that's an endospore, and now you're ready to answer the questions. Okay? So what's this special staining technique called? Endospore. What is the main function of the structure? Seen at the pointer, and it's green. And what is it? No, that the main function, not the primary stain. Survival. Survival. You can't put anything else. That's its function. Okay? You can't say protection. It's not doing that. Right? Protection is like what the capsule does. But survival is, you can't get rid of these things. Yeah? There's a big difference. Okay, now, what is the primary staining reagent in this methodology? Malachite green. And it can't just be malachite, because then you're talking about a stone. Words have meaning. But If you spell it Malcheate, malachite to rhyme with parakeet, I'm still going to count it right because I can phonetically understand what you were trying to tell me. Okay? But if you mean malachite green and you say malachite blue, that's wrong. Okay? Harlow, why are you looking like you're all stressed out? Okay. She's like, okay, so why am I doing this? Well, because each of you all, when you go into your programs, which is surgical technology, nursing, or dental hygiene, you're going to have checkoffs. Checkoffs are oral exams. And I go over there and I talk to those people all the time, and they say, we want time practicals. We want them to be ready to do checkoffs. The first practical, if you're taking the practical in class, you will be able to ask 2 questions at the end of the practical to clarify. But after the first practical, they're all timed and there are no questions to be asked unless you ask questions at the point when we are asking the question. Yeah? Good? I want you to transition slowly. I don't want you to be completely off guard. So I will give you the first one so that you can ask 2 questions. You can't ask more than 2, that's it, 2. Okay? Yes?
I don't know if I missed it. How much time are you going to give us for like one slide?
About 2 minutes. Now, if it only has 1 question, about 30 seconds.
Okay.
But if it has, like if I have several, I'll give you some time. And while we're there talking about that, you can raise your hand and you say, can you clarify question number 6? And I will be happy to. But it's an oral exam. Okay? Everybody with me? Okay. Other things I can ask is, what's the counter stain? And you'll say what? What's the counter stain? Safranin. What's the decolorizer? Not in this one. What's the decolorizer? I heard acetone alcohol.
That's for Gramstein, right?
Yeah, that's for the Gramstein. Water's not a decolorizer, it's a rinser.
Iodine.
Nope, that's a mordant for the Gramstein only. There is none. So see, you all weren't paying attention to me when I was talking about this because I said, I am going to ask you on a practical, what is a decolorizer for endospore stain? And your answer is none. And here you are giving all these crazy answers.
I didn't say that.
What is? Come sit over here. Right here. Yeah, I can't really control that, I'm sorry.
No, I didn't take that.
But we're going to be taking the practical in the classroom next door. And so it's not going to be a problem. Here you go. Test my test. Okay, here we go. Ready? Now, earlier today, because I forgot that I had changed this, there's 2 slides at the very end of this one that is not on the current, that's on Blackboard. I will change that this afternoon because I forgot. I try to make it better every time and I forget. Right? So I will put this on there for you all so you'll have it. Okay, here we go. So again, you're looking at what color? It's what? A Gramstein. Don't get excited yet. It's Gramstein first. What's the purpose of iodine? It's a mordant. That's a really beautiful answer. There are others. What could you say instead of mordant? Sealant, fixer, adherer, all those things mean the same thing, and I'll accept every one of them. I'm very amenable.
So fix it,
Huh?
fix it, can be one,
Fixer.
fix it.
Okay? What's the primary morphology scene? Coccus. If you said coxy, that's fine. But if you say round thingy, it's going to be wrong. Okay? True or false? Again, when you answer true or false, you write the word true or false. Your T's and your F's look the same. True or false? So the bacterium in this field of view is molar. True or false?
False.
Why? It's coxy by definition are all non-molar.
Are we gonna have to explain like in true or false question?
No.
Or just, okay.
It's just the answer. Okay? Good. But the reason I'm doing that is because I want you to understand why I'm asking the question. Okay? All of this is application to everything we've been doing in the lab, which correlates with everything that we studied in lecture. Okay? There's a method to what I'm doing. It's all supposed to be helping you learn. Okay? Here we go. Boom. Okay, looky here. So, what is, in your mind, what do you see immediately? Streak plate. Identify the culture method. It's a streak plate, right? What's the purpose of this method? Be specific. Isolation. All I need, you can write a sentence. The purpose of this is to isolate bacteria. I'm like, you're going to run out of room on your little page. But isolation is all up. Usually, the answers are one word, maybe 3 words. They're not supposed to be a paragraph. Okay? All right, here we go. Identify the object at the pointer. A colony. You can't call it a round thingy. No, because a CFU is different. A CFU is one cell. But my next question says, what is a CFU? And you'll say a colony forming unit or one cell. But Nikki, that's beautiful clarification, right? Because a colony is composed of many cells that came from one cell. That one cell is called a CFU. Okay? Good? Everything we've done in the lab is fair game. Yep. The CFU stands for colony forming unit. It's one cell. But the colony, Rachel, was derived from that one cell. When it landed on the plate, it started to grow, and as it started to grow, it grew into a colony. Which is, everybody's taking their test already, right? Yes. So what on the like the question about the little scenario, it said composed of many cells. That means that they were a colony. And that was one of the things you could have written down. These things are colonial by nature, and only prokaryotes do that. Right? There were like 8 things you could have said. That's just one of them. The most important one and the easy one is the DNA wasn't in a compartment, therefore there was no nucleus. That's the easy one. Right? But there was a lot of them. They're small, they had a slime layer. I mean, there was tons of stuff in there. All you had to do was pick them out, and I just needed 3. There was 8, I just needed 3. Okay? All right, here we go. What tool is used to conduct this method? A loop. A loop is fine. You can say inoculating loop, but a loop is fine. Okay? Why is this going to be hard? You've been doing this the whole time. You've been writing reports the whole time. You've been studying for this. You see what I do? I'm having you do things that's supposed to solidify your understanding of what's going on. Okay, here we go. So what color do you see? And what is it then? A gram stamp. So now, what is the gram specificity and the morphology? And I want you to use 3 letters to answer the question. What are you going to say? GP what? GPR or GPB? Good. True or false, the bacterium in this field of view contains an LPS in its cell wall. False. It's gram positive, there is no LPS. Okay? And I don't need you to explain it. I just need true or false. And you need to write out true or false. All right, here we go. Looky here. So be careful here because these things will get you. You need to look, huh? What's up? It's just for really correct. By definition, it's gram-negative. Good. But this is really looking at something else. This is looking at these things here. So what's the special staining technique? It's a flagellar stain. And by the way, this morning. So is it molar or non-molar? By definition, all spirulli or spirochetes are molar. By definition. So what's the this is the hard one. What's the flagellar arrangement? You fell into my trap. You fell into my trap. Why? Because this one's showing a tuft. These things are you're looking at a three-dimensional organism in one plane. So if you see more than one here, they're in a tuft. That immediately moves them into a different arrangement. What are they? Lophotrichus. Be sure you understand what I'm asking. Just because that's telling you what it truly is. This one of them either broke off or it's behind here and we can't see it because we're only looking at one plane. Okay? Lopho. You want to spell it?
Lophotrichus. So can there be more than one in like both ends?
Yeah.
Okay. I thought it was just. Yeah, I thought it was like one and one. Remember?
And then what's this one? With an L. Sorry.
That was the Peritrichus.
A loaf.
Apotrichus.
It's a loaf of them. No, because then I think one loaf of bread. No, I don't know. A bunch of layers.
So funny, bro.
Okay. Anybody have any questions? I might ask this question. True or false, this organism has an endoflagellum?
No.
That's correct. False. It's not a spirochete. It's just a spirillum. Nice, Justine. That is really nice. Beautiful. Beautiful answer. As we say in science, you're smarter than the average bear. Actually, Yogi Bear said that first. You know who Yogi Bear is?
Yeah. Want to go get somebody?
She's like, I said, Yosemite. Yosemite. Oh, there you go. Bob up.
No, I was saying, like, where's he, like, actually from? I mean, I know where he lives, where he, you know.
All right. What are you looking at?
Your imagination.
Well, you said it's going to be.
Motility media.
Very easy.
So what's the consistency of motility media? It's a semi-solid. What percent agar is in motility media?
0.5.
0.
Good job, Bree. Go, Yogi.
Okay.
That's all you getting from me.
I don't believe that. What tool is used to inoculate the media?
needle.
A needle is fine. You can say needle, you can say inoculating needle. You cannot say long, straight thingy.
Can we draw
You'll be
it?
surprised. No, you can't draw it. I don't want any artistic work. Okay? Identify which tube illustrates a non-molar organism. A. Why?
It's not fuzzy.
I like that. That's fair. But the other one, it's only growing at the site of inoculation. All these are fuzzy. Now, for this class, you can use it's fuzzy or not fuzzy. But don't decide to become a microbiologist and go to some senior level and say it's fuzzy. I'll be so embarrassed if you tell me that. But here, because we're trying to make sense of a fairly difficult and very dense subject matter, I'm okay if you use those things. Okay? Anybody got questions? Here we go. Boom. Okay, so I will have pictures of a microscope. This is just a rendering. But I don't like to ask questions about all the parts of the scope. I only like to ask about the most important part. And the most important part is that guy, these guys, those two knobs right there, and this little bitty lever right here.
Stage.
Huh?
The stage?
No, not the stage, the diaphragm.
Oh.
Okay.
So 9 or 11?
Is it locked? Hey, Guy, are you here to get your homework? Okay. Hold that for a minute. Hey, everybody, say hi to Guy.
Oh, I think you literally were saying, hi, Gay. Mine is recorded, but he doesn't back it up with the class. Well, that's the thing. I need to maybe put it right now.
It was before like anyone else got there, so I guess she saw me alone and she just thought she could join. And I was like, and oh my gosh, I was like, I couldn't do the presentation, but I was not having it. So I was like,
on the discussion board. Okay.
Just 30 minutes, just 30
That like
minutes.
right next to us.
It was weird.
Only when he
Yeah, then
I know, she's like, I just need to go over something.
I don't
She
know
was
if
just
you
sitting there on her phone.
Yeah.
And I was like, right.
And then
I'm sorry, that was a slight intermission because one of my students showed up to pick his homework. They have an exam on Monday, and their exam is juicy. Okay, here we go.
No, I have a question.
Here we go.
Question.
Yes.
Clarify, were you pointing at 9 or 11?
9.
Because you were like, you need to know the diaphragm. Is it the
This
9?
number 11. Number 9 is the stage.
Yeah, I was gonna be like the stage.
Yeah.
But I thought you
Number
had
11.
diaphragm. That's why I was like, wait, did I get that wrong?
Oh, we haven't had the test yet?
Well, no, but like if I did.
Okay. She goes, I wondered if I got that wrong. Like, we haven't had the test yet. Y'all are too stressed out for me. Y'all need to take a chill pill. Okay, here we go. Identify the microscope part labeled 1.
Ocular lens.
Ocular lens is fine. Ocular is fine. No, not just lens. It cannot be just lens because that's its secondary. Secondary function. Its primary function, it's an ocular or an eyepiece. It's bringing the field of view to your eyes. Yeah, eyepiece is good. Huh? Okay. So now, what is the magnification then of this? 10 times. So now, what is the part labeled 4 called? Rotating nose piece, or just nose piece is fine. Okay? Nose piece is fine. Okay, so now these things are called what? Objectives or? Lens. Either one. Now it's lens is okay now, because that's its primary function. So what is the magnification of the oil lens? 100 times. But what's the total magnification?
1,000.
Why is this going to be hard? That's what I'm asking. Yep.
What was 26 again?
You like the 10.
Oh, that's the nose piece.
And 27 is the lens,
Oh.
and 28 is 1,000.
No, 27 is
So I will never ask, well, I shouldn't say that. Okay, so those are the ones that I like to ask most about. The other two are these things right here. What is this little thing here?
the dial.
The dial. No. This little thing right here. Now, this is the big one's, of course. This one is the fine adjustment. And you can't, like, just say knob, or what did you call it? Dial. You can't just say dial.
Fine adjustment.
Fine adjustment is the small one, and the large one is the coarse. Okay? Nikki, why are you making faces at me?
It's like a golf course.
The little one, right there. This little one right here.
Wait, so that's the bottom.
This one down here. This is one of the buttons to turn it on or off.
I think that was 14.
It is.
No, it's because it's similar to this one. I think.
Oh, they're all a little bit different.
Yeah, I think for that microscope, the little ones they're fine too. But the ones we use, it's in the middle of the course.
The little one, no matter on what scope, is always the fine adjustment. The big one is always the coarse adjustment. On the other side of the scope, a lot of times we'll only have the fine, not the coarse.
So if you were to like be like, what's number 10? You would have
That's
to say
the coarse adjustment.
Like course, like COURC?
C-O-R-S-E.
Oh, okay.
Yeah, but you know, Christy, if you said C-O-U-R-S-E, I would accept it.
Okay.
But that means like a course like we're taking a course.
Or golf course.
Yeah, okay. Yeah. Okay? You play golf? You don't play. Here we go.
I do. I say golf.
Okay, these are Nikki's favorites. Okay? So this goes back to the first and second day, right around the time of the big freeze. And I said there are 3 different places where bacteria originate from. And I said if it's really large and rough, they come from soil. If they're small to medium and brightly colored, they come from people or animals. And if they are watery looking, like they're moist looking, they come from water. So now, what is the origin of the structure labeled A? Water. It looks like it's a drop of water. What is the origin of the structure labeled B? Soil. Yep, it's that easy.
So what would be like? Is there like a person?
Yeah. So the little tiny white guys or the yellow ones, those are all from people or animals.
Because they're not like they don't look like.
No, because they're brightly colored, and almost all things that are brightly colored coming from people. Mm-hmm. In a clinical setting, okay, if you go and do environmental work, the rules go out the door. Okay, you have to make your own rules. Okay, here we go. So now, how many different species are on this plate? This is a very good question. No, that's how many colonies. If I ask how many colonies, it's 8. Well, let's count them. I think it might be 9.
4?
Let me count them first. 1, 2, 3, 4, 5, 6, 7, 8, 9. So if I asked how many colonies, it'd be 9. However, I said how many species. So this one, and that one, and that one, and that one, and that one are the same. That's 1. That's 2, that's 3, that's 4, and that makes it 5.
4.
So Cook said if the organism's colonies are similar, they're the same. If they're different species. Okay? Good?
What's the last one you're pointing at?
This little tiny dude right there.
So
Rita,
who's
you want to
4
use my
on
glasses?
accident?
You're like,
Who said 4 on accident? You-
I
Like you're
probably
pointing at
would
the red?
accept 4. Because that one is really tiny, it's hard to see. If you said 4 or 5, I'd be good. Okay? She's very funny.
That's who I am.
Okay, so I just have one, but I'm not getting how these are like different, the same, or what.
So,
Are they the same?
Robert Cook said, if the colonies look different, they're different species, and that is absolutely true. This one, that one, and that one are all the same. That's different, that's different, that's different, and that one's different. So 1, 2, 3, 4, 5. No, that doesn't have any true shape. Those are just crystals of something. This one has a nice round shape. The others don't, right? They're all kind of goofy looking, right? So they're smudges, and they're probably going to be salt crystals. Okay?
But if you don't identify that rule, you're okay with that.
Yeah. Guys, this is a review. I promise you, if I was to ask something like that, it would be definitive. There could be no other answer except the right answer. Okay?
That's how you usually run the tests just in general?
What's that?
That's how you usually run the tests just in general?
The practice?
And all of the other tests?
No, the other ones are open to interpretation. So somebody today wrote something that I had never considered, and I don't remember what. Does anybody remember who was that said something that? Huh?
The periplasmic?
Yeah.
membrane?
So I asked, where are the hydrogens going? And you wrote the periplasmic space, which I had never thought about. I thought plasma membrane. But that's right, because that's where they end up. And I was like, what are you talking? And then I read, oh. I said, well, actually, that's true. So I am open. When I am grading these things, I am trying to discern what you're trying to tell me. And if it makes sense, you're going to get credit. Okay? That's the first time I've ever thought about, and I've used that question before. Right? But I've never really thought about that. But you're the first one who brought it up. So that's good. You're thinking like a scientist. Like it. Okay, here we go. Boom. Okay, so now this is broth. Okay? This is broth, just plain broth, right? So what is the term used for the growth pattern in eggs? Turbidity or turbid? That's correct. If you said cloudy, I would frown and probably, but I'd give you credit because that's what it is. Except that's just not a scientific term. Cloudy means you're out with your boyfriend or girlfriend, you look up at the clouds and you're all romantic. Oh, it's all cloudy today. Yeah, okay. It's different. Okay? What is the term used for the growth pattern stimuli? Can I finish the question first? Color. And then what about this thing down here?
Okay.
So, okay, good. Okay, now what am I looking at?
Wait, can you say it again?
Which one? Which one? 34? That's a polyp.
You guys, one of those.
Yeah.
Not a pool, a polyp.
At the bottom, that's, yes, that is, yeah.
All right.
No, can you go back to, I don't think we did.
You didn't do 32.
Yeah, that was the origin of the colony.
Oh, okay. What is it? Oh,
That's a pointer.
I would have to
Is it
take that
water?
because it's orange. To me, it's from water.
Oh.
But listen, again, you guys are bringing up things I didn't consider. So to me, if that's orange like that, if you said animal, it's acceptable. Okay? You okay? You're making faces at me. Okay, here we go.
So is that from water or?
Either one of those is correct. Because usually when it's kind of shiny like that, it's from water. But because it's got like an orange color to it, I would accept people or animal. All right, here we go. Boom. Okay, we did that, right?
Yes.
Okay. Here we go. Okay, so now what are you looking at? No, it's not a capsule. It's a negative stain. It's not a capsule because you don't see any purple in it, right? So, the staining technique is a negative stain. And what is one advantage of doing the staining? True morphology is perfect. No distortion is fine. Okay? Here's another question. Is this culture pure? Is this culture pure?
No.
And it's not. Why? Because it's not the same organism. And by Cook's rules, if they look different, then they are different. Okay? So you got this guy, you got that guy, you got the little tiny guy, and you got these little coxy looking dudes. So there's at least 4 organisms here. This is what happens at the end of the lab with Dante. That's why I always tell you put all of your organisms in the baskets and we don't use them because they get so contaminated after a class. Even though you're getting better and better, you're still sloppy. Okay? Here we go. So what is this thing called? Not, if you put Tupperware, it's gonna be wrong. This is the anaerobic container. Okay? Now, I teach at different campuses. Here we use this. No, gas pack is something different. This is an anaerobic chamber. This is an anaerobic jar. I use these at HLC when I go over there. Okay? But let's just stay here because this is what we use here. So, is when set up correctly, does this container contain CO2?
Yes.
No. It is completely anaerobic. There's no oxygen in there. So there can't be any CO2. CO2 comes from the liberation of the organisms aerobic respiration. These are anaerobes. Okay? All right, so name a type of bacterium that would grow in this container. There are 3 of them based on their need for oxygen. The easy one is what? Okay, that's the hard one. Facultatively anaerobic would grow. That's the hard one. The easy one is what? An anaerobe. The other one that would grow is what? An aerotolerant organism. Okay? The 2 that would not grow are what? The 2 types of organisms based on their need for oxygen that would not grow in this container are what? Aerobe and microaerophile. Microaerophile still needs a small amount of oxygen.
Wait, so is this the lab that we didn't do?
This is the lab we didn't do.
Oh, because I'm like,
We talked about it in
wait,
class.
I remember you were
I
showing
drew
us, but
things
I
up there. You took a CS today.
Oh, you could, okay, okay. What would be the third thing for growing? I only have 2.
What did you have?
Anaerobic organism, aerotolerant organism.
Facultatively anaerobic. Facultatively anaerobic.
Facultative, can we just switch it? We have to go.
That's not a good thing.
Facultative.
Okay, so I know facultative is hard to spell. This is the same thing, and this is used throughout microbiology. So if you want to use that, I'm perfectly okay.
What is that?
Facultatively anaerobic. I know, you guys are like, I took that's the null hypothesis. I'm taking statistics, that's the null hypothesis. In microbiology, this means anaerobe, and F in front of it means facultatively anaerobic. Okay? What that means it can live with or without, but it prefers oxygen. Aerotolerant, I am saying facultative. It's not like faculty. Okay? All right, here we go. We're almost done. Boom. Okay, no. Okay, looky here. So we've learned purple and red is what? Green and red is what? And now hot pink and blue is what? Acid fast. So what is found in the cell walls of the fuchsia bacteria that make it resistant to staining and contribute to its dangerousness? It was written on the board in the other room when I came in. It started with an M, and Rachel said, "What is that? Is that part of this test?" I'm like, "No, mycolic acid." Mycolic acid. If I ask, "What is the primary stain?" you'll say what? Carbofusion, or fusion, or basic fusion. If I ask, "What is the decolorizer?" you're going to say what? Acid alcohol. If I ask, "What is the counter stain?" you're going to say what? Methylene blue. There are only 2 staining techniques that use decolorizer. The ground stain used what? Acetone alcohol. The acid fast stain used what? Acid alcohol. Okay? All the others do not have decolorizers. This is thioglycolate. Now you have to think about it differently. This is pre-reduced medium. What does that red stuff mean? What does the red stuff mean? Will you go over there and grab Chris? He's looking for me.
And in fact, that actually was not one of the originals, dude.
It was in the back.
Yeah, it is.
The clearance.
And a little.
I need a red one, I need a lap, I need
I can't take a picture.
The other thing I need to. No, my breakfast was my Red Bull.
I could, yeah, I
I'm
could
like,
have
so hungry.
We have it.
I don't know how next Tuesday we're gonna have a lecture after because I'm gonna be so.
In the lab? Are you in the lab? Oh, the lecture is lab one.
Well, last time is gonna be lecture.
Okay, I'm back.
That's
Okay,
what I'm saying.
so we're looking for the types of organisms that are based on their need for oxygen. This red area means that there's dissolved oxygen. Nowhere else in that tube except for the red is there oxygen. So if the organism grows at the top of the tube, what type of organism is it? An aerobe. If an organism grows away from the top of the tube, only at the bottom, it's what? An anaerobe. If the organism grows throughout the tube, but there's more growth at the top of the tube, what is it? That one grows in the middle, Doc, and not at the very top, at the not very bottom. Okay? So if it grows throughout the tube, but there's more growth at the very top, what is it? It prefers oxygen, but grows with or without it. That's facultatively anaerobic. And if it grows throughout the tube equally, it's an aerotolerant organism. Yes, anybody? Questions?
Wait, so what's the difference between B and C? It's just like, is that orange at the top?
Yeah, but there's no difference. They're the same growth pattern.
Okay, yeah, that's
Like, what is throughout the tube again?
Which throughout the tube equally? Aerotolerant. Okay. The orange thing is an indicator for DO, dissolved oxygen. In this case, it's reassuring, but we just basically say that's where the oxygen is. Okay?
And then the organism that can grow with or without is the facultative.
No.
No.
Grows with or without, but prefers oxygen, so it grows more at the top than anywhere else.
Okay.
That's fact. Huh?
I can spell it backwards.
So you can just look at it like this. Look, you want me to? Okay, I'll spell it.
It's not backward.
Like the FO.
I'll make my baby take a nap.
Oh, there's
Okay.
something right. Yeah.
Here we go. Okay, I guess we're done. So I didn't. What was missing that I didn't put on here?
All of those.
What?
That last slide was
And the anaerobic dark.
Oh, I know. I told you all.
Oh, you're right, in the very
I
beginning.
was gonna fix
Very
it,
beginning.
but I didn't have a capsule stain on here. Capsule stain's still gonna be asked about, maybe. I never know what I'm gonna ask until I get into testing.
We're
Right?
gonna ask about the biohazard.
Oh yeah. So 10% of the questions, so that's 5 or 6, are gonna be about safety and lab management. So if I ask, where do you put plates after you're done with them so they can be destroyed? You can't just point them
The
away.
red bag.
The red bag.
No, but that's.
The red biohazard bag. I would rather you say biohazard bag. If you say red bag and I don't know, somebody has a red bag that day, I'm gonna get all confused. Okay? What if I say, what's a permissive temperature? Body temperature. But if I say, what's a permissive temperature in degrees Celsius? 37. First day of class. Okay? What if I say, where would you put plates where you want them to grow on the red tray? All of that is fair game. I might ask, name the 4 things that are in your personal drawer. Oh, you can't open it up and look at it.
Burner, the
Bunsen burner,
little
the little what?
this thingy. This thingy.
A rack,
A rack, and then
The lens
oh,
paper.
the thing,
the what?
The lens paper.
Lens paper, and what's that
and then
thing?
this thing.
The fire
What's that?
starter.
It's not a fire starter.
Fire maker.
What is it called? Lighter?
Lighter. Light it up.
Striker.
It's the striker.
Strike.
We're gonna call it by what it is.
It is.
I mean, it is a fire starter. It
What if you put fire?
So is Drew Barrymore in the movie.
He's like, no, wrong.
Okay? Anybody have questions? Everything that we've done in the lab is fair game on the practical. That's why I call you up and talk, and most of you aren't paying attention. But now you will. Because now you understand. So on Tuesday, the practical will start at 10:30. I will give you probably till 10:40 to get here. And then I'm gonna go. Okay? Next Thursday, and then afterwards we'll do control. And then next Thursday, I will finish control, and then the lab is all about biochemistry. And there's 6 things that you have to write about for a pre-lab. Okay? You don't have to do it now, but just know it's gonna get more technical. Okay? I think we're done. Done.
7 3 doctors next